Today's topic is biomolecules. In this lesson, I'm going to tell about a brief introduction of biomolecules, different types of biomolecules in cell and their functions, and carbohydrates, basic functions, and different types of carbohydrates and their chemical roles and biological roles. Biomolecules are mostly organic molecules which are present in living organisms. So these molecules are naturally occurring and which are having essential elements like carbon, hydrogen, and oxygen. Examples for these biomolecules are carbohydrates, proteins, nucleic acids, and lipids. And these biomolecules, which are present in cell and which are having many features, and it has different shapes and dimensions. Most of the biomolecules are in the form of monomeric and polymeric forms. Polymeric forms, example polypeptide in proteins and polysaccharides in carbohydrates. So these biomolecules, which are present in living matter as well as non-living matter. In living matter, carbon is most abundant element, and non-living matter, oxygen is most abundant element. So why? Because here 46.6% is occupied by oxygen and 18.5% is occupied by carbon in living matter. Biomolecules can be divided into two important types: inorganic biomolecules and organic biomolecules. Organic biomolecules are having the essential elements of carbon, hydrogen, and oxygen. Examples: carbohydrates, lipids, amino acids, proteins, enzymes, nucleotides, nucleic acids, and vitamins. Whereas in case of inorganic biomolecules, it doesn't have carbon element. Examples: minerals, gases, and water. And biomolecules can also be divided into two types based on size and molecular weight. The first type is micromolecules and second one is macromolecules. Micromolecules are smaller in size and having low molecular weight, that is 18 to 800 Dalton units. Whereas in case of macromolecules, which are larger in size and having more molecular weight, that is more than 10,000 Dalton units. Micromolecules are acid soluble, macromolecules are acid insoluble. So these are the examples for micro and macromolecules. There are four important basic biomolecule molecules are present, like proteins, DNA, RNA, polysaccharides, and lipids. And these biomolecules are having major functions in the cell, and these biomolecules are having basic building blocks, like proteins are having amino acid as a building block, and these amino acids will be sequenced and forming a polypeptide chain. Here the protein will be useful for the structure and function of the cell. DNA can be useful for hereditary information, which carries the genetic information. RNA, which is having ribonucleotide, a building block, and which is useful for the protein synthesis. Polysaccharide is having a monosaccharide as a building block, and it is useful for the storage of energy. And the lipids, which are having fatty acids and glycerol as building blocks, and which are useful for the energy for the long-term demands in the body. And next, I'm going to tell about the carbohydrates. The word carbohydrate, which is obtained from hydrate decarbone, which is a French term, that means hydrate of carbon. A carbohydrate which is having many carbon atoms which are having hydrogen and hydroxyl group. And these carbohydrates are having the essential elements of carbon, hydrogen, and oxygen. But the ratio of hydrogen and oxygen will be 2:1. Examples for carbohydrates are sugars, starch, and fibers. Yet these carbohydrates are the most abundant source of energy. That means it is giving 4 calories per gram. And these carbohydrates are also available in the form of glycoproteins and glycolipids, which are useful for the cell membrane synthesis. And it is also available in the form of cellulose in plants, exoskeleton in some insects. In humans, it is available in the form of glycogen to meet the energy demand of the body. And these carbohydrates are mainly used as the precursors for the synthesis of fats and amino acids. And if you are consuming more amount of carbohydrates, it may give complications, health problems like obesity and type 2 diabetes. Carbohydrates can be divided into three subparts: monosaccharides, oligosaccharides, and polysaccharides. Monosaccharides are the basic units of carbohydrates as simple sugars, which are again divided into two types based on number of carbon atoms and also based on the functional group. Oligosaccharides, which are crystalline in nature, water soluble, and sweet in taste, they can be easily hydrolyzed. It is also again divided into three important saccharides: disaccharide, trisaccharide, and tetrasaccharides. Disaccharides are having two monomers of monosaccharides, and trisaccharides are having three monosaccharides, and tetrasaccharides are having four monosaccharides, which are held together by a glycosidic bonds, and polysaccharides, which are having many repetitive monosaccharides, which are held by glycosidic bonds. But these polysaccharides are non-crystalline in nature and water insoluble and tasteless. Example for polysaccharides are starch and cellulose. Monosaccharides are divided into two important types based on carbon atoms and based on functional groups. If the sugar is having three carbon atoms, it is called to be triose sugar, 4 tetrose, 5 pentose, 6 hexose, and 7 carbon is giving heptoses. So these are the examples of different monosaccharide sugars. And the next part is based on functional group. Here the sugar is having aldehyde functional group, then it is called to be aldose sugar. If the sugar is having keto functional group, then it is called to be keto sugar. Example for aldose sugar is glyceraldehyde and glucose. Example for keto sugar is dihydroxyacetone and fructose. Derivatives of monosaccharides. So these monosaccharides or simple sugars can be derivated by because it is having hydroxy group and functional groups like aldehyde group and keto group. There is a change in these functional groups. It may leads to formation of derivatives. In deoxy sugars, hydroxy group present at second position, at second carbon, is absent. That means there is no hydroxy group at second carbon. That is called deoxy sugars. In amino sugars, a amine group is present, so which is replacing the hydroxy group in the sugar. That means a hydroxy group will be replaced by amine group, then it is called to be amino sugars. In sugar acids, aldehyde and alcohol groups get oxidized and forming an acid group, then it is called to be sugar acids. In sugar alcohols, aldose or ketoses, that means which are having aldehyde functional group or ketone functional group, so which gets reduced and forming alcohols. So these are the deoxy sugars. Here at second position of this sugar, so this sugar is called ribose sugar. Why? Because it is having 5 carbons, 1, 2, 3, 4, and 5. At second position, hydroxy group is present in D-ribose sugar, whereas in case of 2-deoxy D-ribose sugar, here at second position it doesn't have hydroxy group. In amino sugar, hydroxyl group will be replaced by amine group. Here actually the hydroxyl group has to be there, but here amine group is replacing the hydroxyl group. So then it is called to be amino sugars. That means the sugar which is containing amine group. Example, glucosamine and galactosamine. So these are acid sugars which are containing acid functional group. Example, beta D glucuronate and D glucuronate. Here the glucuronate is in beta position. So that means here carboxyl group is present which is going upward direction, and also here hydroxyl group which is present at this carbon is also going into the upward direction. This is the cis form of sugar, so that's why this is called to be beta sugar. If hydroxyl group is present in downward direction, that means a trans form gives alpha anomeric forms. So these are the examples of acid sugars: beta D glucuronate and D glucuronate. Here D glucuronate is a linear molecule. And the next one is sugar alcohols. Here aldehyde group is getting reduced and forming alcohols. So glucose is reduced and forming alcohol, that is called sorbitol. So here the examples for sugar alcohols are sorbitol and xylitol. And the next one is oligosaccharides, so which are having 2 to 9 monosaccharides which are held together by a glycosidic bond. So these oligosaccharides can be divided into three important saccharides: disaccharide, trisaccharide, and tetrasaccharide. So these are the examples of these saccharides. Disaccharide is having sucrose and lactose. Trisaccharides, example raffinose. Tetrasaccharide is having stachyose. But in which disaccharide is most commonly used one. So these disaccharides are having two monosaccharide units which are held together by the glycosidic bond, and these disaccharides are crystalline in nature, water soluble, and sweet in taste. These are the examples of disaccharides: maltose sugar, lactose sugar, and sucrose sugar. This maltose sugar is also called to be malt sugar. In this malt sugar, two glucose molecules are held together. Why? Because it is a disaccharide. In lactose sugar, so which is also called to be milk sugar. Why? Because it is present in the milk. It is useful for the storing of the milk. Here this lactose sugar is made up of glucose and galactose. And sucrose, which is also called to be cane sugar and table sugar, which is having two monosaccharide units like glucose and fructose. But this sucrose is the main important source of dietary carbohydrates. The next one is polysaccharides. So these polysaccharides are also called as glycans. These glycans can be divided into two parts: homoglycans and heteroglycans. Homoglycans are made up of one type of monosaccharide monomers. For example, if you are taking all glucose monomers are held together and forming a long chain polysaccharide, then it is called to be glucan. If the long chain polysaccharide is having fructose monomers, then it is called fructan. If the polysaccharide chain is having galactose, then it is called to be galactone. So that means homoglycans are having only one type of monosaccharide monomers. But whereas in case of heteroglycans, here it is having two or more than two monomeric monosaccharides are present. So these are the examples of heteroglycans like hyaluronic acid, agar, chitin, and peptidoglycans. So these polysaccharides are having a repeated units of monosaccharides and which are held together by glycosidic bond we have already discussed. And these polysaccharides doesn't produce any sweet, it is tasteless, and it is useful for the storage purpose only. And this polysaccharide is also divided into storage polysaccharides and structural polysaccharides. Example for storage polysaccharides are starch, glycogen, and insulin. Example for structural polysaccharides are cellulose and chitin. So these are the examples of storage polysaccharides: starch, glycogen, insulin. Starch, so which is a carbohydrate reserve in plant and which is present in cereals, roots, tubers, and vegetables in high content. So that is why it is very much important dietary source for the animals. And this starch is having monomeric glucose, glucon is present. And this starch is also having the combination of amylose and amylopectin. So amylose is a monomeric polymeric chain, which is having all glucose molecules are held together by glycosidic bonds. And this amylopectin, so which is having a side chain, it's a branch for this amylose. So this is amylose, so which is having many glucose monomers, and these glucose monomers are held together by a glycosidic bond that is called alpha 1,4 glycosidic bond. So why? Because here fourth carbon of one glucose molecule is attached with one carbon of another glucose molecule. So this is alpha 1,4 glycosidic bonds. So this straight chain or linear chain is also having a branching. So here this branching is attached with this sixth carbon. So that's why this is called alpha 1,6 glycosidic bonds. If branching is present, then it is called to be amylopectin. If branching is not present, then the molecule is called to be amylase. So that means here the starch is containing amylose and amylopectin. And the next one is glycogen. So glycogen is having a carbohydrate reserve in animals. So that's why this is called animal starch. So which is present in liver, muscle, and brain cells. So which is also present in plants, but in few parts. So it is not present, like chlorophyll, and it is also not present in yeast and fungi. And this glycogen is also having a monomers of glucose, so which are held together by a glycosidic bond, forming a polysaccharide chain. But there is a difference between this glycogen and starch. In starch, a branching is present. In glycogen also branching is present. But here glycogen, so branches are present at every 8 to 10 monosaccharides, at every 8 to 10 glucose molecules. But whereas in case of starch, here the branching is present at every 15 to 20 molecules of glucose. So that means here the branching is intensified in glycogen. So less branching is present in starch in comparing with glycogen. The next one is insulin. So this insulin is having the polymer of fructose. So many fructose molecules are held together and forming insulin. So this insulin is found in bulbs, garlic, onion, etc., and which is water soluble. That's why it can be filtered by a kidney. So this insulin will not be metabolized in our body. It can be filtered by a kidney. So that's why it is useful for kidney functioning. And the next one is structural polysaccharides. Here, these are the examples for structural polysaccharides: cellulose and chitin. Cellulose is mostly abundant in plants, especially in cell walls. It is useful for the cell growth, and it is not available in animals. Chitins are also having second most abundant organic substances, so which comes under heteropolysaccharides. So this chitin which is present in especially in invertebrates, so which is having exoskeleton, invertebrates like insects and crustaceans. And this chitin will be providing strength and elasticity to the skeleton, but this is not having more strength. So when this chitin is impregnated with calcium, then it gets hardness, then flexibility will be decreased, elasticity will be decreased when hardness will be increases. And finally, the carbohydrate summary. Here the carbohydrates are polyhydroxyaldehydes or ketones, which is having aldehyde function group and ketone function group, and many hydroxyl groups are also present. And these carbohydrates can be divided into mono, oligo, and polysaccharides, as we have already discussed. Here the glucose molecules, which are the simple sugars which are available in the form of alpha and beta anomers. I've told you alpha anomers are transforms, transanomers, and beta anomers are cis forms. In carbohydrates, so disaccharides are most commonly used sugars. Example, malt sugar, milk sugar, and table sugars. And polysaccharides can also be classified into homo and heteropolysaccharides. So these polysaccharides are water-insoluble non-crystalline materials. In homosaccharide, only one type of glucose monomeric is present. In heterosaccharide, two or more than two monosaccharides are present. And this carbohydrate, which is very much important biomolecule in our body, and it is giving instant energy to our body. If you're consuming more amount of carbohydrate, so it leads to few complications like type 2 diabetes and obesity. So this is the end of the topic. Thank you.